What I want to do is just introduce you a little bit to university teaching, which is the whole point of you coming here, so you get a bit of an idea what we do. Remember that if you came here properly, you'd have hour lectures instead of half-hour lectures, so you'd have some boring old fart like me going on for an hour rather than the 25 minutes that I've got now. But what I want to do is just talk to you in the way I would talk to undergraduates, basically, about biochemistry, and one particular aspect of biochemistry, and that's the biochemistry behind sex. And I want to look at the hormones that make you either males or females, and I want to look at some of the chemicals in the environment that might interfere with those hormones, and that are causing quite a lot of changes in populations at the moment. So we go from the basic background of biochemistry and sex all the way through to the rather more complex areas associated with hormones. So let's have a bit of a look first at sex. If I could just find my pointer. You'll notice that the person on the left is me, and the reason... Why do you laugh?
Because he doesn't have glasses.
Oh, thank you. That was a very good answer from the front, because he doesn't have glasses. I love you, and if you were at this university, you just got a first, I'll tell you that now. So you'll see this is a man, and you can see it's a man very easily, even though I've covered up the naughty bits. And the naughty bits are how you would really tell it's a man. Then on the right-hand side, we've got a lady, and I've covered up two lots of naughty bits because there are two ways of identifying whether something is a lady or not. But those two sets of naughty bits, or those three sets of naughty bits, are determined and designed in the biochemistry of you when you're in the embryo. And those naughty bits, their design was created initially by just a few cells being converted into different sorts of cells. And those cells were converted because of particular hormones that were interacting with them and making them either make this very handsome bloke here or this very ugly woman here. Is that biased or what, do you think? So let's just have a look at those hormones a little bit more closely. So I've got two hormones here, and I've covered up their naughty bits because we can't have you exposed to the differences between men and women, can we? So on the left-hand side, this is a symbol for woman, as you probably know, and this is a symbol for man. And on the left-hand side is a molecule called 17-beta estradiol, and you probably know it as estrogen or estradiol, and that's one of the female hormones. There are three, but this is one of them, the most active of them. And the one on the right is the male hormone testosterone. So all the women in the audience have got a whole bunch of this hormone, and all the men have got a whole bunch of this. But the men have got a small amount of the female hormone, and the women have got a small amount of the male hormone as well. And it's the ratio between these two hormones that causes your cells to grow in a way that you're either beautiful by this lady that says exactly the right things, or ugly like this bloke here who hasn't said anything and didn't deserve that at all. You can thump me later, and I've got this feeling you're going to. Yes. Yep, he's going to thump me later. So I'm going to need some testosterone later because that also helps in the fighting reflex. So look at those two molecules, and you can see that they're pretty well the same. I've just left a little tantalizing little bit of difference there. You don't need to understand the chemistry of these molecules, you need to understand their shapes. But if any of you are chemists, you can look at the chemistry in your mind at the same time. So we've got a ring structure here with 5 members. We've got a 6-membered ring, another 6-membered ring, and something else behind there. And here we've got exactly the same. They've both got a hydroxyl group at the top, and they've both got a methyl group there. This one doesn't have a methyl group in that position, this one does. The hydroxyl groups, I think you probably know that triangular sign means that they're coming out of the page. So they're sterically arranged coming out of the page. That's very, very important. If they went into the page, for example, if that went into the page, it would not be the female hormone. It would not work as the female hormone. You'll see why in a few minutes' time. So I think what I'm going to have to do, I'm going to have to uncover those censored bits. Now, I was going to uncover them on the last pictures, but I was told I wasn't allowed to. But is there anybody under 18 in here? Oh, well, you just have to close your eyes because I can't let you see what I'm just about to show you, because this, ladies and gentlemen, is the difference between men and women. Oh, my God. Are you excited? Yes. Would you like to look at those for a few seconds, just to get accustomed to the differences? Let's have a look at those differences then, shall we? On the left-hand side, we've got a hydroxyl group on a benzene ring. It doesn't sound as exciting, does it? A hydroxyl group on a benzene ring. And on the right-hand side, we've got a keto group on a 6-membered ring with a single double bond. Those are basically, apart from the methyl group in this position, are the only differences between those two molecules. You've got to admit it's bloody amazing that that determines whether you're a male or a female. You'd think if you were going to design that system, you'd make them a bit more different, wouldn't you? But that's not the way it worked. Evolution created those two molecules which are sufficiently different to allow our bodies to recognize them. But how do our bodies recognize them? Who's heard of receptors before I go on? Most of you, I think, will have heard of receptors. So receptors are big protein molecules that float around in the blood or sit in cells, usually sit in cells, and they can recognize other molecules. They recognize molecules by the shape of that molecule. So the molecule they're recognizing is like a key, and the receptor's like a lock. If the key fits the lock, it switches on the receptor, and the receptor does all the things it's going to do, which we'll talk about in a minute. So in a sex context, there are several receptors, but I'm just going to talk about one of them, because I haven't got enough time to talk about them all. The receptor is the estrogen receptor, which is represented here as the orangey thing. Here's a steroid hormone. In this case, it's estrogen. It comes into the cell. It interacts because its shape is exactly right to interact with the shape of that receptor. And when it interacts, it forms a receptor-ligand complex, and that receptor-ligand complex changes shape very slightly. We'll look at that in a minute. Extremely clever, that tiny difference in shape. Because of that tiny difference in shape, it can interact with an area on DNA. It's called the estrogen responsive element. It doesn't matter about that, but I'd like you to know that I know what I'm talking about. I'm not sure I do. And when it interacts with that, it switches on a whole load of genes. And the genes it switches on are all the genes that go to make a female-female. If this was the testosterone receptor, or the androgen receptor as it's called, and this is testosterone, it works in a very similar, a little bit different actually, but a very similar way, and it does exactly the same thing, but it switches on all the things that make men men. It switches on the ugliness gene, for one. And I know because I've got a whole load of that one. So once we've got the hormone, you're producing the proteins that determine that cell's sex, basically. So let's have a look in a little bit more detail, but schematically, how this happens. My research group does a lot of this work, and we're at the moment looking at how molecules interact with that receptor, and I'll show you some of that in a few minutes' time. So let's look at it schematically at the top here. Look, we've got the estrogen receptor, that boxy black thing, and on the top, I've got the binding cleft as a sort of triangulary two fingers sticking up thing, and then estrogen itself as a triangle. It sits into that receptor, and when it sits in that receptor, it changes the shape of this molecule. I should have shown it changing shape, actually. And when it changes shape, it means it can bind to another one. So two of these bind together to form a dimer, and when they form a dimer, there's an area at the bottom that then interacts with DNA, and that then upregulates the genes. Very clever. So it makes it almost foolproof, because it requires the structure of that molecule to be exactly the same. So the testosterone molecule doesn't fit into that receptor. It can't activate it. It can't switch it on. So therefore, only estrogen, in theory, can switch it on. So let's have a look at that in a little bit more detail. Here's the actual human estrogen receptor that one of my PhD students created in a program called Schrödinger. Took a whole year to do this, putting each individual amino acid in at one at a time, doing all the thermodynamics to see how they would fold, and ending up with exactly that molecule. In it here is estrogen, and we know exactly where estrogen binds. We know exactly where it binds because we can use techniques like X-ray crystallography. We can make a crystal of the estrogen receptor with estrogen in it, and we can see exactly where it is. And there it is sitting in there. But it'd be really nice to know how it binds. And of course, we can work that out too. And all we need to do, although it takes years to do it, is to basically magnify that bit here where all these individual amino acids in the protein are sitting and see how they react or how they interact with that estrogen molecule. And here it is. So there's that bit magnified, and you can see from that magnification, there's estrogen, and here are all of the coils of protein structure, and we can see where it sits, but we can't see what it's binding to. we can't see what it's interacting with. So we need to magnify it even more, and we can do that too. So here it is, the ultimate magnification now. So we've got just that binding bit of the protein magnified to see exactly what happens. So here's the estrogen molecule, and we know it forms a hydrogen bond, the dotted yellow line, with histidine, which is one of the amino acids in the protein structure. And we know it forms a hydrogen bond with glutamate and arginine, two other amino acids. And we know it needs a single water molecule to allow that reaction to occur. Now, when it binds in that way, and this is the really clear, I get very excited about this stuff. Don't get bored yet. This can be really exciting. The bottom part of the molecule, first of all, binds here, and then the top is too far away from this to bind, and so it pulls it down. And when it pulls it down, this helix here moves. Boom. And when it moves, it changes the structure of the whole protein to allow the two proteins to bind together. And that's called a conformational change. And the only reason the whole thing works like that is because this molecule is designed, in quotes, to fit exactly into that binding site. Clever, clever stuff. We can really simplify that down to the simple requirements of that key, the estrogen molecule, to fit in to the estrogen receptor. So here it is, look, here's the estrogen molecule. This is my representation of the estrogen receptor. It requires a hydroxyl group at one end on a 5-membered ring ideally, or an aliphatic ring, i.e. one without a pi cloud for the chemists amongst you. And on the left-hand side, it requires another hydroxyl on a pi cloud 6-membered benzene ring structure. And the distance between that one and that one has to be, not should be, has to be 12.1 angstroms. Now we're getting really specific. If it fulfills those criteria, then it will bind, then it will go to DNA, and it'll switch on all the feminization of the cell. Just to remind you, looking at testosterone and estrogen again, the difference is that and that. That does not form hydrogen bonds. So that tiny difference means that it won't hydrogen bond with the receptor, therefore it doesn't switch it on. Just as a little aside, it does switch it on to 3%, a tiny, tiny bit, but not significantly. And you can superimpose those two molecules. I like doing this. You ready? Boom. I'll do it again backwards. I'm very happy when I managed to do the computing necessary to do this. There you go. So you can see that it lines up absolutely, except that methyl group and the difference between those two groups there. So let's explore that a little bit more and just look at other molecules. So we've got two molecules, we've talked about them, testosterone at the top and estradiol or estrogen at the bottom, and we've got a molecule in the middle. Before I go any further, and if anybody does, I'll be really disappointed because it completely steals my thunder. Does anybody know what this molecule is? Did you know? Don't tell anybody, please. Looking at that molecule, does it, put your hands up, does it look like testosterone? Hands up. Does it look like estrogen? Yeah, I mean, you're on the ball here. It doesn't take much. You don't need to be a Nobel laureate chemist to realize that. It looks like estrogen. So if I gave this to somebody, you'd think it would fit into the receptor because it's got the hydroxyl in the right place and the right separation. Everything's identical, except it's got this group here, which is an ethanol group. Apart from that, it's exactly the same. And indeed it would. If I gave you a dose of that, it will bind to your estrogen receptor like you've never seen. It binds incredibly strongly. I'll tell you what it's called, because I like names of chemicals. It's called ethanol estradiol. Does anybody know what that is? It's the contraceptive pill. And it was designed absolutely to fit into the estrogen receptor so strongly that it stops estrogen binding. And when it stops estrogen binding, that means you can't get pregnant. It disrupts the estrous cycle. So it stops the estrous cycle, stops all the biochemistry necessary to make you pregnant. And the only reason, the only way that was created was to design it understanding how estrogen binds to the estrogen receptor. And that is a process called molecular mimicry. So one molecule looking like a natural molecule that has properties very similar to the natural molecule. Let's look at a few more. Ethanol Estradiol, the pill, was designed, it was made to look like that, but there have been a few accidents. So let's look first of all at the female hormone, 17-beta estradiol, how we should really look at it in 3 dimensions. Because in 1 dimension, it doesn't quite tell us enough. So we need to understand the 3-dimensionality of it. When you see that and see it in this form, it's difficult to envision that as that. But you can see, look, there's the hydroxyl group, this one, it's around the wrong way actually, and there's that hydroxyl group. Sorry, this is around the right way. There's that hydroxyl group on the end here, and there's that hydroxyl group on the end there. But they're almost at 90 degrees to each other. So to bind to the receptor, they have to be in that shape. So if any other molecule is going to look anything like this, like ethanol estradiol, it has to be in the same shape. I'm not going to show you all of those. So just keep all that in your mind. Then we'll just look at something that might appear to be completely different, but I'm going to try and connect the whole lot together in a few minutes' time. So a whole load of things are going on in the environment at the moment. This is a trout, a male trout. Male trout are producing a protein called vitellogenin. Vitellogenin is the protein that's used to make eggs. Male trout should not produce that because they are males. They don't have estrogen, and it's at very low levels, and it's produced in response to estrogen. Alligators in America have shorter penises now than they had many years ago. The same might be happening to humans. There is an experiment here that we're not going to do. Because I get told off when I do those sorts of experiments, and you're smiling an awful lot. You would like us to do it. Later. And that was actually shown by a chap called Louis Guillet, and Louis just died actually a few months ago, which is very sad. And he literally, with his PhD students, which are expendable human beings, went out, caught alligators, measured the length of penises over a 10-year period, and showed they declined. Oysters aren't breeding as well as they should, and the human sperm count is going down. Why? And the reason is estrogen. But more than that, all of these processes that I've shown on here, I didn't mention this one, this is precocious puberty. Girls are coming into puberty earlier now. The average age has declined by 2 years over 1 decade. It's amazing. All of these things are controlled by estrogen. If the alligator's penis is getting smaller, it means that the ratio of estrogen to testosterone has gone up. There's more estrogen than testosterone. Puberty in girls is initiated by them producing estrogen at a particular age, normally around about 11.5 to 12.5, when the process starts biochemically. Male fish would not produce vitellogenin because they don't have enough estrogen. It's only produced in females. So why are all of these creatures responding in this way? Let's have a look at a molecule. This is called bisphenol A. It's a monomer of some plastics, polycarbonate plastics. It's the stuff that's used to make your spectacles as well, my glasses. The glass is actually polycarbonate plastic. It's also used in many other places, which we'll talk about in a few minutes' time. But look at that. Does it look a bit like estrogen? It's got 2 hydroxyl groups, and it's got a benzene ring, and it's got a separation. If I tell you the distance between that and that is 12.1 angstroms, you suddenly get quite interested. And if I tell you that the 3-dimensionally it's identical to 17-beta estradiol, even though it doesn't look like it, you get quite interested, or you should do. Let's have a look at it. You can superimpose them very nicely, and they line up beautifully. And in fact, bisphenol A combine to the estrogen receptor and cause all the effects that estrogen causes. Here's some uses of it. It's used to line tin cans, like baked bean tin cans. You sometimes see a white lining on the inside, that's bisphenol A. It's used to make these lovely new fillings that we all have now, which replace the horrible ones, some of which I've got, and it leaches out. And when it's manufactured, it leaches into the environment and contaminates the environment. So if the alligators, if the fish, and if we are getting small doses of this, which believe me, we are, we found it in milk in my lab yesterday, then we could be having binding to our estrogen receptor, slowly converting ourselves to a more female form. Let's have a look at the theory behind that, though. You can take bisphenol A, you can isolate the estrogen receptor. This is work over years and years. You can put them together. You can make a crystal, which takes a very long time, and these are tiny crystals 1 nanometer across or 2 nanometers across. You can do X-ray crystallography. You can solve that crystallograph, and you can show that bisphenol A binds to the estrogen receptor. It binds at the bottom here, just in the same way as estrogen does, but it doesn't bind at the top. Slightly wrong to bind at the top, but for the chemist amongst you, the electronegativity of this hydroxyl interacts with the electropositivity of part of the histidine molecule, and it just pulls it in that direction. It never forms a bond, but it just pulls it down. And it pulls it down just enough to change the conformation of the protein, cause the dimerization, go to DNA, and produce feminization. We've shown that in my lab. It's very easy to show. So that's one molecule. Let's quickly have a look at a few more. Does anybody recognize that? Don't expect you to, really. I like you not to, because then I can tell you. That's soy. And soy is used in lots of foods. It's used to make soy milk. I'll talk a bit more about where else it's used in a few minutes' time. And soy contains a molecule, lots of molecules actually, but one in particular that we're rather interested in. And there's some other uses of it. Soybeans themselves, these sort of crackery things, soy milk, or a soy curd. And the molecule that it contains is a molecule called genistein. Look at the structure of that. You're getting good now. You're looking at that and you think, I know why it's shown us that. It's got a hydroxyl group here. It's got a hydroxyl group there. You never guess. It's 12.1 angstroms between that and between that. There's a lot of other gunk in the middle that we don't like. But it binds to the estrogen receptor. It switches it on. It does just the same thing. So if you've got a dose of that because you eat a load of soy, then you will have that problem too. Let's just have a look at those two molecules. The red one is 17-beta-estradiol. The blue one's genistein. You superimpose them. Look at that. They line up beautifully. If you do it in 3 dimensions, it's even better. But it's hard to see that. Who's eaten soy today? Put your hands up. So one person. Anyone else? Who's eaten that today? So you've all eaten soy because all New Zealand bread contains soy. This might be a big government push to convert everybody into women as a feminist approach to life. I don't know. But you've all eaten soy. And the point I'm making is we're all exposed to these chemicals every day for every part of our lives. And therefore there's a feminizing pressure going on at the moment. And if you're exposed to these individual compounds, the effect is tiny. But if you're exposed to mixtures of them, they all have the same effect on the same receptor. You can add up all of these effects. And my research group has recently shown that these effects are absolutely additive. And so the change in sperm count, the decrease in alligator penis length, the decrease in the multiplication rate in oysters, and the production of proteins in fish are almost certainly all related to this. So right from that initial understanding of the biochemistry of sex, we can come all the way round to some of the environmental effects that we're seeing currently, which are based on molecules that look like hormones and mimic hormones. And they're called xenoestrogens from the Greek word xenos, which means foreign. So I hope that's given you a little bit of insight into the sort of research we do here and the sort of stuff we teach. I have a whole course in this. We have 12 lectures on it actually, and it's a fantastic course that you'd have to take. So thanks for listening. If there are any questions, now's your time. Thank you. Thanks so much.